We are approaching a collapse that modern medicine cannot stop. By 2050, 10 million people a year are projected to die from infections that drugs can no longer touch. But the answer to this crisis isn't hidden in a multi-billion-dollar laboratory. It's hanging from the trees you walk past every day. Before the first pharmacy existed, people didn't just survive. They possessed a biological technology that has been perfecting itself for 400 million years. This is the story of Usnea, the medicine that was so effective it had to be forgotten, not because it failed, but because it was impossible to own. This is the story of one of those places, one of those medicines, and the long, quiet, deliberate process by which it was taken from you. There is a plant growing right now within a few miles of wherever you are sitting. It hangs from tree branches like gray-green thread. It drapes over old fence posts. It colonizes the north-facing sides of boulders in damp forest clearings. Its common names read like a vocabulary of deliberate neglect. Old man's beard, witch's hair, tree's dandruff. Names designed, whether intentionally or not, to make sure you keep walking past it without ever asking what it actually is. Its scientific name is Usnea, and inside its pale, thread-like structure, it produces a compound called Usnic Acid, a molecule so precisely calibrated for the destruction of bacteria that it has been doing so, without fail, for approximately 400 million years. Long before humans existed to observe it, long before we had the language to describe what we were seeing, long before anyone thought to ask why the old healers packed it into wounds, brewed it into teas, and passed the knowledge of its use from generation to generation with the same quiet seriousness they passed down the knowledge of fire. To understand what was lost, you have to walk into a world that operated by different rules. Not worse rules, different ones. Rules written in the accumulated, hard-won, generationally tested experience of people whose survival depended on getting it right. The Roman legions bled on 3 continents. A wound left untreated for 48 hours in a warm climate could kill a man more reliably than any sword. The military physician Dioscorides, writing in 65 AD in De Materia Medica, documented treatment for infected wounds using what his Greek text calls the hanging growth of trees. That book remained the foundational pharmacological reference of Western medicine for 15 centuries. In all that time, the knowledge of Usnea never entirely disappeared. It simply became inconvenient. Viking warriors packed lichen into wound dressings before crossing the North Sea. Archaeological burial analysis confirmed it. They called it sky thread, ghost wool, and believed it carried the immunity of the forest into the wound. They were wrong about the mechanism. The outcome was real. In China, Usnea appears in herbalist records under the name Songluo, prescribed specifically for the lung conditions that modern medicine classifies as bacterial pneumonia and bronchitis. Protocols refined across centuries of observed outcomes, without a single laboratory, without a single microscope. The Cherokee used it, the Ojibwe, the Haudenosaunee. Indigenous nations across North America, with no contact with Rome or China or the Norse, arrived independently at the same conclusion about the same plant treating the same illnesses. Different continents, different languages, different cosmologies. The same answer. The researchers at Oldways Lab call this Convergent Healing. When multiple unconnected human civilizations across different continents and different millennia independently identify the same plant as a solution to the same medical problem, that convergence is not coincidence. It is not mythology. It is evidence. The same kind of evidence that underlies all medicine, the observation of outcomes. across the entire inhabited world, for thousands of years, without coordination, without anything connecting these cultures except the fact that they were paying attention to what worked. But how? How can a simple lichen, ignored by modern doctors, destroy pathogens that the strongest hospital antibiotics cannot? The answer lies in a microscopic war. A mechanism of death so perfect that bacteria have failed to evolve against it for 500 million years. Let's look at the data that pharmaceutical companies don't want to fund. Now follow Usnic acid into the interior of a bacterial cell, because understanding the mechanism is the key to understanding why what Usnea does is so remarkable, and why the bacteria we are most afraid of today have so little defense against it. Usnic acid is produced exclusively by lichens. You will not find it in any flowering plant, any tree, any fungus. It is unique to the lichen kingdom, and its exclusivity tells you something critical. It was not a general-purpose compound that happened to be antimicrobial. It was evolved specifically and precisely for the destruction of bacteria in environments where bacterial colonization would otherwise be fatal. Lichens live exposed, on rock faces, on bark, in environments with no shelter, no immune system in the mammalian sense, no capacity to flee. They survive because they make themselves chemically hostile to the organisms that would kill them. And Usnic acid is the primary weapon in that arsenal. When Usnic acid encounters a bacterial cell, it penetrates the outer membrane with minimal resistance. It is lipophilic, chemically attracted to fat, and it moves through the lipid bilayer the way heat moves through metal. The bacterium registers nothing, no alarm, no response. The compound is already inside before any defense can form. Inside the cell, Usnic acid launches its attack on energy production. Every living cell generates energy through a process called oxidative phosphorylation. Protons are pumped across the membrane, creating a gradient that powers ATP synthesis. ATP is the universal energy currency of life. Without it, nothing works. Protein synthesis stops, cell wall maintenance stops, reproduction stops. Usnic acid is a proton ionophore. Think of it this way. Most antibiotics try to break a single wire in the bacterial engine. Eventually, the bacteria learns to bypass that wire. But Usnic acid? It creates a massive short circuit. It shuttles protons across the membrane outside of the normal channels. It forces the bacterial power plant to run at 10,000 RPM in neutral. The bacterium continues burning its energy stores, but the energy releases as heat instead of being captured as fuel. The cell generates immense heat, uses up all its fuel, and literally burns itself out from the inside. It's not just an attack, it's a metabolic meltdown, all combustion, no motion. The cell runs its metabolic engine in neutral until the reserves are gone. As ATP levels collapse, the bacterium loses the capacity to maintain its cell wall. The proteins responsible for wall synthesis stop functioning. The wall begins to degrade from within. In gram-positive bacteria, Staphylococcus, Streptococcus, Clostridium, the thick peptidoglycan wall becomes a structural liability. The cell swells, ruptures, dies. But this is only part of what the compound does. Simultaneously, Usnic acid disrupts DNA replication, dismantles the biofilm, the protective polysaccharide matrix bacteria build as a collective defense against antibiotics, and depolarizes the cell membrane through a secondary mechanism that strips away yet another layer of the bacterium's survival infrastructure. Multiple simultaneous attacks, through multiple independent pathways, all at once. This is what makes resistance so extraordinarily difficult to develop. Antibiotic resistance is evolutionary. A bacterium carrying a mutation that provides even slight protection survives while others die, reproduces, and passes the mutation forward. Given enough generations, and bacteria reproduce in minutes, a resistant population emerges. The resistance timeline for penicillin after clinical introduction was approximately 3 years. For methicillin, less than 2. We now face strains of Staphylococcus aureus resistant to vancomycin, which was designated the antibiotic of last resort. The last resort is no longer the last resort. To develop meaningful resistance to Usnic acid, a bacterium would need to simultaneously evolve protection against energy disruption, membrane penetration, cell wall degradation, protein synthesis interference, and biofilm dismantling. Not 5 variations on one problem, 5 fundamentally different problems requiring 5 different evolutionary solutions at the same moment. The probability is, by current estimates, vanishingly small, and the empirical evidence confirms the mathematics. 400 million years of sustained evolutionary pressure. The bacteria have not succeeded. The compound still works, essentially unchanged. The documented antimicrobial spectrum is not theoretical. Peer-reviewed laboratory data shows significant activity against Staphylococcus aureus, including MRSA, against Mycobacterium tuberculosis, against Helicobacter pylori, against Pseudomonas aeruginosa, against multi-drug-resistant strains that are currently killing people in hospital wards on every continent. Published, replicated, accessible, and almost entirely ignored. The researchers at Oldways Lab did not arrive at this subject through idealism. They arrived through data, through the structural recognition of why the evidence was being ignored. Not because it was weak, but because of what acting on it would mean for the economics of modern pharmaceutical development. Here is the problem. Developing a new drug costs $1 to $2 billion. That investment is recovered through a patent, a legally enforced monopoly lasting approximately 20 years. Without it, competitors produce the same drug the moment it is approved. The patent is not incidental to the pharmaceutical business model. It is the entire foundation of it. Natural compounds cannot be patented. You cannot own what nature already made. This is where the tragedy becomes a strategy. If nature provides a cure for free, it is a direct threat to the quarterly earnings of a corporation. You cannot patent a forest. You cannot put a barcode on a lichen. So, the system does something worse than banning it. It makes it invisible. It labels it a weed and ensures it stays out of the medical textbooks, which means no mechanism exists to fund the trials that would bring Usnic acid through regulatory approval. Any company that paid for the research would immediately find its data used by competitors who spent nothing. The economics are simple, and they are a death sentence for any medicine that cannot be chemically transformed into something ownable. This is not a conspiracy. It requires no secret meetings, no individual villain. It is far more disturbing. A system working exactly as designed. An incentive architecture built around patentable chemistry, structurally incapable of delivering natural medicines to people who need them, regardless of how strong the evidence becomes. The structure is not broken. It is functioning perfectly. And in functioning perfectly, it has produced a world where 400 million years of antimicrobial efficacy cannot find institutional support because it cannot generate institutional return. The World Health Organization estimates antibiotic-resistant infections currently kill 700,000 people annually. By 2050, that figure is projected to reach 10 million, exceeding global cancer mortality. The last genuinely novel class of antibiotics reached clinical use in the 1980s. We are fighting a rapidly evolving adversary with weapons 40 years out of date. Into this landscape, consider again what Usnea represents: a compound with demonstrated activity against the exact pathogens that are currently killing people in hospital settings. Growing freely, on trees, in every temperate forest on the planet, without manufacturing, without supply chains, without cold storage requirements, without a patent number, without a price. The ancient healers who packed it into wounds on Roman battlefields did not know what Usnic acid was. They knew only what generations of observation had confirmed: that this gray-green thread helped wounds close when they would not otherwise close, that fevers broke. They worked with mechanisms they could not name, but they worked with reality, with actual compounds interacting with actual pathogens in actual bodies. And their results were documented for 2,000 years without interruption. That knowledge is not a museum piece. It is part of the evidence base of human health, undervalued, underfunded, sidelined by economic forces that have nothing to do with its efficacy, but still present, still growing on the trees outside your window. The tragedy is not that we forgot. The tragedy is that we were helped to forget. That a system built around patentable compounds had every incentive to keep unpatentable alternatives poorly studied and invisible to the people whose health depended on knowing what was available to them. The researchers at Oldways Lab are in the business of memory, preserving what was known before the forgetting, documenting preparation methods and safety records with the rigor the evidence deserves, ensuring that when the post-antibiotic era arrives in full, and the evidence strongly suggests it will, a body of knowledge remains intact, accessible, and ready, not as a replacement for the pharmaceutical tools we have, as the insurance policy we should have been building all along. 400 million years of evidence, documented across every inhabited continent, growing freely on every temperate tree, ignored not because it doesn't work, but because no one can own it. The plant is still there. The post-antibiotic era is not a maybe, it is a when. And when the pharmacies go dark, the only medicine that remains will be the one that never needed a patent to work. At Oldways Lab, we are reclaiming that memory, because one day your life might depend on the gray-green thread you almost walked past today. The vault is open. It's time to remember. Subscribe to stay connected to the source.